Hello everyone, I'm Chang Lu, a PhD student in Massachusetts University. I'd like to give you a poster presentation to talk about the identification of the sex-specific genes, transcription factors, and signaling pathways in monocytes associated with cardiovascular disease. Cardiovascular disease is a major cause of morbidity and mortality for both men and women, and such differences have been reported both in its clinical manifestation and the underlying pathogenesis. For example, women are more likely than men to die from heart attack because although chest pain is a common symptom for both men and women, women are also more likely to have other less recognized symptoms, such as shortness of breath, jaw pain, and dizziness. It is as yet unclear to what extent these differences are mirrored by differences in transcriptional makeup in monocytic cells, which are instrumental in cardiovascular disease progression. In this study, we comprehensively dissected sex differences in gene expression profiles and regulatory contexts of monocytes obtained from a cohort of cardiovascular disease versus healthy subjects. First of all, I will show you the workflow of this study. In order to study the sex difference, we first integrate microarray data from the Center for Translational Molecular Medicine circulating cell cohort and Academic Medical Center cohort in Amsterdam. Here we show the name of these two cohorts as CTMM and GSE 98 and 20, and then split the whole cohort into female and male groups. Both of these two groups were then performed the same computational pipeline. Subsequently, we compared the analysis outcomes between females and males to find the cardiovascular disease-related sex difference. For the first step, we merged the CTMM and the GSE 98 and 20 cohort for increasing the number of healthy controls. Both of these two cohorts are Illumina microarray data, which were extracted from CD14 positive monocytes of cardiovascular disease patients and healthy subjects after a series of pre-processing procedures such as transformation, normalization, and outlier detection, in total of 496 samples, including 342 males, 154 females were used in this study. We then split the merged cohort into female and male groups to dissect the sex differences in cardiovascular disease from downstream gene expression to upstream pathway in monocytes. In this step, we first performed the differential gene expression analysis using lemma on female and male separately. The cardiovascular disease-related gene expression signatures, or GES for short, from male and female subjects would then become the inputs of the procedures of pathway and transcription factor activity inference. Next, female and male specific pathway were identified based on the stability of pathway activity enrichment score. It was calculated based on a predefined pathway response gene coefficient matrix from progeny, which recorded the weights and the significant levels of a gene to 14 dominant pathways. Similarly, we compared the activities of transcription factors on female and males by energetic ranked-based enrichment analysis called AREA and Dorothea, which is a resource containing directional transcription factor and target interactions for analyzing sex-specific transcription factors driving these sex-specific GSs. Subsequently, we applied Carnival to extract the regulatory network architectures for both men and women GSs from a prior knowledge-based protein-protein interaction network called OMI-PATH and predict their upstream regulators' activities guided by the most dysregulated pathways and transcription factors we inferred from the previous steps. We identified sex-specific pathways by studying how the adjusted p-value cutoff from progeny coefficient matrix will affect normalized enrichment score on our female and male subjects. Here we call normalized enrichment score as NES for short. Most pathways kept basically stable with an increase of minus log adjusted p-value for both female and males, which indicated the validity of progeny. KF-couple and TNF-alpha show significantly or almost significantly stable positive NES on females, but not on males. And EGFR and PI3K are significantly inhibited only on males. From the result of TF activity inference, we identified 20 female and 20 male-specific transcription factors, as well as 9 sex-independent transcription factors with statistical significance, according to the difference between NEIs of transcription factors on female and male cohort. For example, HNF1A is significantly activated on female subjects but not on males, but IRF3 is inhibited as a male-specific transcription factor. We also generated a female and male signaling pathway network based on the female and male-specific pathway and transcription factor activities. As we can see from the result, for instance, HNF1A as a female-specific transcription factor is indirectly activated by ACTL6A and inhibited by SOX17. In conclusion, we comprehensively analyzed the sex difference in monocytes associated with cardiovascular disease by identifying and comparing the female and male-specific gene signatures, transcription factors, and signaling pathways. These sex-specific pathways and transcription factor will be validated on multi-ethnic study of atherosclerosis cohort dataset. In the end, thank all of the co-authors' contribution to this study, and thanks for your listening. If you are interested in my work, feel free to talk with me anytime during this conference. I will be glad to meet you online.